Hello everyone, my name is Beatriz. I'm an in vivo pharmacologist, scientist at Therapeutics, a biotechnology company with headquarters in US and offices and laboratories in Switzerland and Spain. It is my pleasure today to present at the Society for Neuroscience one of the GAIN's latest findings showing the positive effects of our lead compound named as GT2287 for the treatment of some of the main pathological features of Parkinson's disease. GAIN focuses on protein misfolding diseases, including neurodegenerative diseases. For this, GAIN develops medicines that target novel allosteric binding site on misfolded enzymes using its patented CTX discovery platform. This is a computational platform that uses 3D structure of enzymes and a computational technology to discover new allosteric binding sites that can be targeted for therapeutic benefit to correct misfolded enzymes, promoting protein maturation and eliminating toxic substrate build-up that cause the disease. Parkinson's disease is a neurodegenerative disease affecting millions of people worldwide. 5 to 10% of all Parkinson's patients have a GBA1 mutation. Mutations in the GBA1 gene, which encodes the lysosomal enzyme Glucocerebrosidase, also called GCase, are responsible for the build-up of glucosylceramide, involved with alpha-synuclein pathology in Parkinson. PD patients with GBA1 mutations cannot be distinguished at the clinical level from idiopathic PD patients. However, PD-GBA patients have an early onset of the disease, faster progression, and non-motor symptoms. It is important to mention that GCase deficiency activity levels have been shown to be disrupted in PD patients carrying GBA mutations, but also in sporadic PD patients, highlighting the importance of GCase as a target for all PD patients, broadening the therapeutic window. Abnormal GCase lead to a reduction of alpha-synuclein degradation in the lysosome, with subsequent alpha-synuclein accumulation. Since there is a bidirectional feedback loop between GCase and alpha-synuclein, increasing alpha-synuclein oligomers lead to further decreasing normal GCase activity, which in turn promotes formation of additional alpha-synuclein oligomers, exacerbating the disease. GAIN targets dysfunctional wild-type and mutant GCase with GT2287, which specifically binds to GCase trapped in the endoplasmic reticulum, promoting its stability and folding. GCase then avoids its degradation, which translates into more enzyme trafficking to the lysosome. In the lysosome, GCase has an improved activity, along with the restoration of normal lysosomal autophagic function, and ultimately decreasing alpha-synuclein levels. As a small molecule, GT2287 can be orally administered. It has also an excellent brain penetration, and importantly, it has a good toxicity profile. To test the ability of GT2287 in reducing some of the main pathophysiological features in a Parkinson animal model, we first decided to use the very well-known and characterized Rotenone induction model. This model reproduces important features of the human disease, including loss of dopaminergic neurons, increases phospho-alpha-synuclein, as well as behavioral and motor deficit. Rats were treated with rotenone as well as with GT2287 at 3 different doses for a 7-day period. And day 8, locomotor function and rearing behavior were tested. In addition, immunosuppression of phospho-alpha-synuclein, EVA1, and tyrosine hydroxylase were also determined in the substantia nigra and striatum, respectively. We showed that after 7 days of oral treatment with GT2287, 60 mg per kilogram induced a significant reduction in phospho-alpha-synuclein, as well as in microgliosis in the substantia nigra, compared to rotenone rats without the drug treatment. In addition, we show increased dopaminergic TH positive cells in the striatum after GT2287 treatment. Importantly, 60 mg of GT2287 show a tendency towards locomotor deficit improvement compared to rotenone rats without the drug treatment. Both 60 and 90 mg significantly improved rearing behavior in rotenone rats compared to those without the drug treatment. Finally, we can see here representative traces of locomotor activity in the open field of a vehicle-controlled rotenone and rotenone-treated rat. We can conclude that the orally bioavailable and brain-penetrant lead GT2287 ameliorate alpha-synuclein pathology, inflammation, and behavioral phenotypes in a dose-dependent manner in a Parkinson rat model. This finding points to the lysosomal GCase enzyme as a promising target for the restoration of key biological activities found to be impaired in GBA-related Parkinson disease, as well as in sporadic PD patients, thus guaranteeing further development towards the clinic. I would like now to thank you to the great team integrating Game Therapeutics based in Bethesda, Lugano, and Barcelona.